This video is going to be on mitosis and meiosis. So the cell cycle is the time from when a cell is first born to the time that it undergoes cell division again. And the cell cycle is made up of two primary stages, interphase and then M phase. M phase stands for mitosis, and interphase includes three main important steps. So first of all, after that cell is first born from the previous mitosis, it enters into G1. G stands for gap phase, and this is a time when the cell undergoes just growth. Occasionally, a cell will make the decision that it's not going to divide again. It's going to withdraw from the cell cycle, and that is shown here. That is called G0. After G1, if that cell has decided to go through with another cell cycle, it will then enter into S phase. S stands for synthesis, and that is the time during which the DNA is replicated. So DNA replication is a process that we're going to talk about in much more detail later in the course. After S phase comes G2. So G2 is preparation for mitosis, and then the cell enters into mitosis, and two main things have to happen. First of all, the chromosomes themselves actually divide and separate from each other, and then secondly, cytokinesis occurs. Now, I'd just like to make the point that meiosis is also preceded by the very same type of cell cycle. Meiosis, as we'll talk about in a minute, consists of two cellular divisions. However, we don't actually do a meiosis, do a whole cell cycle, and then do a second division of meiosis. The way it occurs is that there's only one cell cycle preceding meiosis. So we have one S phase, one time that the DNA is replicated, and then another G2 phase, and then we do meiosis 1, followed immediately by meiosis 2. Now I'd like to say a little bit more about what happens during S phase. So in eukaryotes, we have linear chromosomes, and they have a couple of key parts to them. There's a region called the centromere, which does not necessarily have to be at the center. Sometimes it is, sometimes it isn't. And then we have the telomeres, which are the ends of the chromosomes. So during S phase, that DNA molecule, that's one long DNA double helix molecule, gets replicated, and then at the end of S phase, you have two DNA molecules. Now, these DNA molecules are still attached to each other, and they attach to each other at the centromeres by a structure called the kinetochore. These two DNA molecules are identical to each other, and they are called sister chromatids. Now, one important point here is that as long as those two sister chromatids are attached to each other, they are still considered one chromosome, as it's shown down here. Let's talk about the stages of mitosis. So as a cell gets ready to enter mitosis, just remember that this cell has already undergone S phase, and so the sister chromatids are already present at that point at the end of S phase. So mitosis has 4 steps: prophase, metaphase, anaphase, telophase. Some sources, including your textbook, call prometaphase as a separate phase. I'm not actually interested in having you distinguish prometaphase. What we will do is all the events that are spoken about in prometaphase, we're going to call just prophase or late prophase. Okay, so starting with prophase, the nuclear envelope starts to break down. The DNA condenses so that you can actually visualize those sister chromatids. And by late prophase, these pairs of sister chromatids begin to be captured by the spindle fibers that are emanating from the poles of the cell. During metaphase, the chromosomes line up at the equator of the cell, which is also called the metaphase plate. At this point, each chromosome has 2 spindle fibers, one going to each spindle pole. And during anaphase, then, the kinetochore breaks down, and the 2 sisters separate from each other. One gets pulled to one pole, the other gets pulled to the other pole. And this happens separately for all of the chromosomes. In telophase, the chromosomes reach those spindle poles and the nuclear envelopes will start to reform and the DNA begins to decondense. At the very end of telophase, the cell will then undergo cytokinesis, and splitting the cytoplasm is just, dividing it into two. So to summarize the important points of mitosis, first of all, mitosis is asexual reproduction, just the division of a cell, and this occurs in somatic cells, and a somatic cell is pretty much any tissue that is not a germ cell, which is an egg or sperm producing a gamete. And the products of mitosis are two cells, and those two cells are genetically identical both to each other and genetically identical to the mother cell that produced them. Okay, let's talk about meiosis next. So meiosis is what we really care about most in this course, because this is how we actually do inheritance from one generation to the next generation. This is sexual reproduction, and the process of meiosis is actually the production of gametes, egg and sperm, which are haploid cells. And then that is followed, of course, by fertilization, which is a joining of two gametes together to form a zygote or to form the next generation. The overall advantage of meiosis is that we have genetic variation introduced during the process of meiosis and fertilization. We'll start off with an overview of meiosis. So meiosis consists of two stages, two cell divisions, meiosis 1 and meiosis 2. And as we mentioned before, those are preceded by one interphase. So there's one S phase where the DNA is replicated, and then we go into meiosis 1, followed immediately by meiosis 2. Meiosis 1 is the step that is the really unusual step. What happens in meiosis 1 is that homologous chromosomes, for really the first time ever, right, they find each other and they line up together. This does not happen in mitosis. Each chromosome was independent in mitosis, but in meiosis, homologous chromosomes find each other. And they line up, and then one homologous chromosome goes one way, the other one goes the other way. Meiosis 1 is also called a reduction division, and the reason it's called a reduction division is because you actually have the number of chromosomes. So in this example, you start out with 4 chromosomes, and you end up with 2 cells. Each of those cells has 2 chromosomes at the end of meiosis 1. In meiosis 2, the sister chromatids separate from each other. So that is very, very similar to events that occur in mitosis. And this is not a reduction division. This is called an equational division. So you start out with 2 chromosomes and you end up with cells that each have 2 chromosomes as well. And each stage of meiosis can then be broken down into the 4 steps: prophase, metaphase, anaphase, and telophase. So if you are looking at the step of prophase that happens in meiosis 1, we're just going to call that prophase 1 to indicate it's happening during meiosis 1. If you were looking at prophase during meiosis 2, you would call it prophase 2. Okay, so now I'd like to go into the steps of meiosis. We're going to start out with meiosis 1. And prophase 1 is a very, very long period of time, and a lot of different things happen. Because it's such a complicated series of events, people have actually come up with these names to kind of break down prophase 1 into different discrete steps. So you see some of those listed here: leptotene, zygotene, etc. I'm not going to have you learn those names, but you should be aware of the events that occurred during this stage of prophase 1. So some of the things are common to the other prophase, in mitosis that you already learned about. The nuclear envelope breaks down, the chromosomes condense. The other thing then that starts happening is that the homologous chromosomes progressively line up with each other. Now again, remember, those chromosomes are already found in sister chromatid form. And so you actually have 4 DNA molecules that progressively line up, and they line up exactly so that the analogous part of each chromosome is directly next to that same part in the homologous chromosome. So this process of lining up is called synapsis. At the end of synapsis, you have what is called a tetrad. Now, you'll see a couple of different names. Sometimes this is also called a bivalent. I prefer the term tetrad, and the reason is because there's actually 4 molecules of DNA, right? 2 sisters of one homologous chromosome have lined up with 2 sisters of the other homologous chromosome, forming this group of 4. This is shown right down here. So you have these 2 sisters in blue lining up with the 2 sisters of the homologous chromosome in red. One other thing occurs during prophase 1 that's extremely important, and that is something called crossing over, which these sisters can actually exchange genetic material with each other. So they've lined up so that the analogous parts are next to each other, and they can actually do this crossing over where you break and you reconnect the strands of DNA. What you can actually see in a microscope at this point is presence of these crossover events, and these are actually called chiasmata. So to summarize the events of meiosis 1, in prophase 1, the homologous chromosomes line up with each other, synapsis occurs, crossing over, etc. During metaphase 1, those tetrads then line up at the metaphase plate. In anaphase 1, the homologous chromosomes separate from each other. This does not involve breakdown of the kinetochore that is holding the sister chromatids together, because it is the homologous chromosomes that separate at this point, anaphase 1. And then in telophase 1, those homologous chromosomes move to the poles and the DNA briefly decondenses, the nuclear envelope forms, and cytokinesis occurs. Meiosis 2 is very similar to mitosis. In prophase 2, the nuclear envelope breaks down, the chromosomes condense, and in metaphase 2, those sister chromatids align at the metaphase plate in both of those 2 cells that have formed from the previous cell division. During anaphase 2, those sister chromatids separate from each other. Okay, so now is when the kinetochore breaks down. And in telophase 2, those chromosomes arrive at the poles, the chromosomes decondense, the nuclear envelope reforms again, and cytokinesis occurs. And at the end of meiosis, you now have 4 cells, and each of them have a haploid number of chromosomes. So to summarize the events of meiosis compared to mitosis here, so meiosis is sexual reproduction. It involves only the gametes, so the egg and the sperm in humans, and the result of meiosis is actually 4 cells. Every single one of those cells is different both from each other and different from the mother cell that produced them. And all those 4 cells are haploid cells as well.